Well, thank you everybody for coming, and thank you for that lovely introduction, Mifi. What a title, right? So let's start by trying to break down this massive title, which puts together so many different things. So, plant, pathogens, and pirouettes. So, you know, plants. What are plants? I think we're all pretty much familiar with plants. So we've all seen, you know, green beings around us all the time. But let's look at what a plant is from a sort of more precise scientific point of view. So this is a tree of life. So our presentation of how kingdoms of life are divided and how they are related to each other. And you can see here we have a time axis which shows us over time what evolved earlier and later, and how we started from Luca, which is the last universal common ancestor, so the very first living cell. And the tree then splits into bacteria and archaea, which are single-celled organisms. And then we have the evolution of multicellular organisms, and we have animals, plants, and fungi. And plants are all the way up there. Now, to give you an idea of how animal cells are different from plant cells, let's look at some basics. So, in general, quite similar. They all are eukaryotic, so they have nuclei where DNA is contained here and there. But one of the main striking characteristics of plant cells is the cell wall. So this is a structure that is outside the cell membrane. So animal cells have cell membranes. Plant cells also have cell membranes. But the cell wall is a rigid structure, which essentially keeps the structure of the cell contained. And that's why plants are still static. The other thing that is present in both, but not quite the same, is the vacuole. So the vacuole is present in animal cells, but it is usually small. It's a membrane-bound organelle that contains liquid, and it is quite variable in its size and how many there are in animal cells. In plant cells, there's always a very, very large vacuole, and it takes up a lot, and in some cases most, of the cell size, and it maintains, with turgor pressure, it keeps that cell membrane attached to that cell wall, so it maintains its shape. And you know, the elephant in the room is the chloroplast. The chloroplast is an organelle, just like mitochondria are, and while plants also have mitochondria, so here we have some mitochondria here and also here, plants have chloroplasts, which is what allows them to get their energy from the sun and do photosynthesis. And it has evolved later on, so plant cells have evolved later compared to animal cells. Now, let's talk about pathogens. So pathogens are not a specific group. They are several types of organisms. So we're thinking bacteria, for example, or insects, viruses, filamentous organisms, or nematodes. So nematodes look a bit like worms, but they're not quite worms, which essentially make, in this case, a plant ill. That's what a pathogen is. Very straightforward. Now, why do we care about plant pathogens? That is, you know, we all know why we care about human pathogens. We all get sick and we really want to feel better. Well, we lose about 40% of agricultural yield annually to plant pathogens and to plant disease. And there aren't many ways that we know how to treat these or how to stop these. And this is currently an extremely big problem because people's livelihoods depend on this, and also how much food the growing population has. So this is a really big problem. We can treat all the diseases we want, but if we don't have anything to eat, we will die. There's not much way around that. So I particularly am interested in filamentous pathogens. So filamentous pathogens are these within the sort of what we're looking at. And there are really two types of filamentous pathogens, fungi and oomycetes. So let's start by looking at fungi. So fungi are up here in that tree of life, so they are multicellular. They're actually more closely related to animals than they are to plants. And this might be striking for a lot of people, which often think that they are actually similar to plants, but they really aren't. They don't have chloroplasts. They cannot photosynthesize. Now, we all kind of know what a fungus is, but spoiler alert, a mushroom is not a fungus. A mushroom is part of a fungus, is an organ of a fungus, the same way a gut is a human organ. All of that is also part of the fungus, and that part is called a mycelium. Now, here you see a more detailed picture of a mycelium. So these parts up here that you see are mushrooms, and all this sort of thread-like structure is a mycelia. Now, if we look at a sort of a schematic of a fungus, so the whole thing is a fungus. And it's very easy looking at this schematic to assume that this is a bit like a tree. So you have the mycelium that are the roots and the mushroom that is a tree. But that is actually not the case. This is a lot more representative of what it is. So the mycelium is the main part, the permanent part of the fungus. So that lives all year round, a bit like the entire plant. And the mushroom is actually also called the fruiting body. So it is just the fruit, and it carries and disperses spores. So a bit like a fruit and an apple with its seeds. So let's zoom into the mycelium. So the mycelium is made of these structures called hyphae. This is an electron microscope image of hyphae. So these hyphae are 10 microns in diameter. That's about 1/10 of the thickness of a hair. So very, very thin. And they grouped together, they form the mycelium. And they are formed by single cell lines. So this is a bunch of single cells attached sort of front to back, and they each have a nucleus. Now, some types of fungi, not all species, actually are aseptate, so they don't have any division between cells, and there's just essentially one very large and long cell with a bunch of nuclei inside it. So the whole mycelium tends to grow, if it's not constricted by anything, it grows in a sort of circular fashion, and it comes from a spore that germinates. And this is what a mycelium looks like in a petri dish. So this petri dish is about that big, and that is kind of what you tend to see, how it grows. This sort of fluffiness is the mycelium. And this is, well, it's not magnified. The picture is not magnified, but of course it's blown up. So this is what you see with the naked eye. Now, let's move on to oomycetes. So oomycetes are also filamentous, but they are absolutely not related to fungi. They were originally thought to be related to fungi, but when their DNA was sequenced, we discovered that they actually genetically are not even close. They diverged way before fungi, and they are not related at all. Now, when you look at them in the lab or on a plate, they also have hyphae, and they look exactly the same. But there are a few key differences, and one of them is their spores. So all oomycetes have these swimming spores that have 2 flagella. Not all fungi have, most fungi don't have swimming spores, some do, but none have diflagellated spores. So this is a key distinction. And they do swim around in this sort of very chaotic, this is real time, this is not accelerated, so that's how quickly they sort of swirl around. And then these spores originate from, this is essentially the fruiting body called a sporangia, and it's sort of the equivalent, the microscopic equivalent of a mushroom. And they essentially burst when they're in contact with liquid and release the spores, and this is what a sporangia looks like under the microscope. These are detached from the mycelia, and you can see the spores inside, and one actually has germinated and it's starting to grow a new mycelium. And the other interesting thing about oomycetes is that almost all of them are pathogens, and most of them are plant pathogens. This is a very interesting niche as they evolved before plants, but nonetheless what it is like. And so most of them do require a host, which usually, and need the plant cells to be able to grow a full mycelium and then repeat their life cycle. Now, the oomycete pathogen that I studied most is called Phytophthora. This is a genus of pathogens which has causes a variety of plant diseases. And I looked specifically at two of these. One is Phytophthora infestans. So I think all of us know the Irish potato famine. So Phytophthora infestans is the cause of the Irish potato famine, and it causes potato late blight, which essentially makes the tuber rot like that, but also the plant essentially rots. And it can also infect tomato, and this is what happens. This is a time lapse of a few days, about a week of a tomato infection, and it just fully rots. And again, no crop protection strategies against this. We can not prevent it. It doesn't respond to fungicides because it's not fungus. And then its awful cousin, the tropical cousin of Phytophthora infestans, is Phytophthora palmivora, which is a generalist pathogen, so it kind of infects pretty much anything, but is currently being a huge issue for chocolate and cocoa. It causes black pod disease. So this, I think a lot of us have not seen what a cocoa tree looks like. So these are cocoa fruits which contain cocoa beans, and this is what they look like once they're infected. So they just fully rot on the inside. And the problem is that these plantations are also a very big investment for the families that use them, that have them, because these trees take years, up to 10 years, to grow and have a productive plantation. And then within a year, Phytophthora palmivora can completely wipe out your plantation and then contaminate that soil for another 10 years. So that can be a really long-term damage to people's livelihood. And again, no crop protection strategy available. Some farmers tend to go and sort of chop off infected parts of the plants, but that can be only a short-term solution, and unless you eradicate the presence, one spore is enough to really destroy your field, which is quite an urgent issue if we still want to have chocolate in 10 years. Now, we know how the infection process works. So once a spore attaches to, so this, imagine, is the side of a leaf with the cells of the leaf here. So the spore will attach, then will germinate, and will form what is called an apressorium. So this is a microscopy image. So the pink are the plant cells, and you can see the yellow spores that are germinating. And then when they form apressoria, they essentially use literal pressure. This was measured. There were experiments done by physicists that measured the pressure, and it can apply such a strong pressure that it makes a hole in the cuticle and then penetrates and grows between the cells, not within the cells. And then it forms these digit-like appendages called haustoria, which essentially are feeding appendages, where it breaks the barrier between the plant cell and the pathogen cell, and it acquires nutrients and pumps other proteins within the plant. And this is what that looks like. You can see these pink blobs are the nuclei, the corresponding to this part of the plant, and then you can see the digit-like appendages growing inside the cells. So how do plants defend themselves? Do we know? Well, we know some. So most of you are familiar with animal immune system. Almost all animals, not all animals, have an innate immune system and an adaptive immune system. The adaptive immune system is what vaccines are based on, and how we can generate immunity and make a person resistant to a disease, for example. resistant is probably not the right word for people. Fran will correct me later. Well, plants only have innate immunity. So there is no real way to make a plant resistant, an individual plant resistant to a pathogen. And the innate immune system in the plant is divided into pattern-triggered immunity and effector-triggered immunity. These two happen at different times and are different levels of immune response. So pattern-triggered immunity occurs before the pathogen enters the cells. And it's based on these receptors, which are called pattern recognition receptors, which poke outside of the cell. So imagine this is the cell of the plant. These poke out and they recognize pumps, which are molecules that are common to a variety of pathogens. So things like chitin, which is present on the outside coat of some fungi, but also of insects. These are recognized by these receptors, and then they activate a signaling cascade, which causes a more mild immune response, usually a Ross burst, which is an excretion of reactive oxygen species which are a bit acidic, essentially, and damage the pathogen. Now, they also recognize DAMPs, which are damage-associated molecular patterns. So essentially, if an insect is chewing on the plant will recognize its own molecules that have been chewed up and send an alert. Oh, I'm being chewed up. I need to react. Now, effector-triggered immunity happens later. That happens once we've had penetration. And effector-triggered immunity is specific. It's specific to a species of plant and a species of pathogen, while PTI is a very general response. So, for example, we could have wild potato resistant to Phytophthora infestans. And how this works is that usually pathogens evolve ways to subvert pattern-triggered immunity. And they do this by pumping these molecules called effectors inside the cell and blocking the immune response, the pump-triggered immune response. But effector-triggered immunity uses resistance proteins, which evolve inside the plant to recognize these effectors and then mount a much stronger defense. So hypersensitive response is a very strong way to prevent a pathogen spreading, which is done by killing a bit of yourself to prevent the spreading, also known as apoptosis in animal terms, or programmed cell death. And this is what it looks like in a plant. So this is a bunch of cells that essentially are dead. And if the pathogen is inside there, it will not be able to spread. And this is what it looks like against viruses, because viruses tend to infect a bunch of different cells, so you will get a lot of little dots. Now, what happens is that this, over an evolutionary time span, we will get continuous production and evolution of new effectors from the pathogens, which subvert, again, this, the plant's ability to respond to them. And the plant, in turn, will evolve continuously new resistance protein to prevent the pathogen from escaping its immune response. And this leads to what is called the Red Queen effect, which is taken from Alice's Adventures in Wonderland. So this idea that you need to keep running and keep evolving to stay still, because you need to keep evolving new and new proteins to be able to maintain this equilibrium between resistance and infection. But all this information comes from leaves, and that is a problem. So very few people study roots. However, 90% of those plants that are lost to pathogens are lost to root pathogens. And nobody looks at roots. There is a reason why. Roots are in soil. That's very hard to look at. It's very hard to study things that are inside this very opaque matrix. You need to be able to somehow extract it and look at that. And not only that, but there is so much going on in soil. You have different species of plants interacting with each other and interacting with a variety of different species of beneficial and pathogenic interactors, and in different soil types with different contexts. That is a lot of complexity, and scientists like simple things. So this is a lot to handle. And also, interactors exist on a spectrum. So they exist on a spectrum from mutualism, in which both interactors benefit from the interaction. So this is an example of rhizobia, which is a type of bacteria that forms these root nodules in legumes. And these bacteria essentially produce nitrogen, fix nitrogen, and give nitrogen to the plant. And the plant, in turn, provides carbon to the bacteria. So this is a very good relationship for both of them. And then on the other side of the spectrum, you have pathogenic interactions like the Phytophthora one that I showed you before. So we get infection from a filamentous organism. And now that's even more complicated when you insert context and the presence of multiple of these organisms. So it is known that if a positive interactor like arbuscular mycorrhiza, this is a type of fungus that is actually beneficial, colonizes roots and is beneficial to these roots. If that is present in a plant, then a pathogen like Phytophthora will really struggle to infect. And all of this is also further complicated by the type of soil you're in. So different types of soils will, for example, a more acid or a more basic environment might affect how this interaction occurs. Well, let's simplify this. Do we know how Phytophthora spores find a root? We sort of do. So Phytophthora spores are released from sporangia when water, usually irrigation water, touches the sporangia. And then they swim towards the root and they attach essentially here to the tip of the root. When they are stuck there, that's when they have attached, and they do this process called encystment, and then from there they will germinate. Now the question is, can we actually stop them from finding the root? And this is really what I care about and what I want to do in life. Look at this. So I'm going to tell you about how I helped look into this during my PhD, which is only a little part of the work that I hope to do. Now, this is a V slide. It's a little contraption about that big that I built during my PhD, and it allows me to put things in the middle here in a liquid and apply an electric field. Now, if we put Phytophthora spores in there and we apply an electric field, they all swim to the positive pole. And we thought, well, what if we try and use this? Can we prevent spores from attaching to a root by driving them away using an electric field? And what we're really trying to do is essentially subvert this very first part of the infection process. And that's why I built this other contraption. This is 3D printed, and you have your little roots here, and then you have an electric field applied to the side. And what you can see here is the spores, which are yellow, really do attach and infect the root when there is no electric field, but they don't when there is an electric field. And we also quantified that, and you can see that it's quite effective. So at this point, we wanted to make sure that this helped reduce the overall infection and not just the first part of the infection. So essentially, if we reduce the number of the spores that attach here, do we actually get less growth in the plant? And we quantified the plant growth, sorry, not the plant growth, the pathogen growth inside the plant, and the answer is yes, it does help. It actually really, really helps. Now, you probably thought I forgot, but we're also talking about pirouettes. So a pirouette is a ballet step in which the dancer turns around a few times, hopefully 2, 3 times around itself, and within a ballet. So probably I should now say that I am a ballet dancer as well, and I've been a ballet dancer for a very long time. And something that I've been asked repeatedly, I want to say in interviews as well, is what do science and ballet have in common? And the answer to that for me is storytelling. So science tells a story. We spend years and years looking at all the details of how something works, and then we describe it in a scientific paper. This is my first author paper for my PhD. I have quite a few now, but that was special. And ballet is another way to tell stories, usually more of a human story. Here we have an example of Romeo and Juliet performed by Royal Ballet. And if ballet is so good at telling stories, can we use it to tell a science story? Well, that's how the SciBallet Project was born. So I currently run the SciBallet Project, and we've been able, well, with the help of quite a few people, bring 3 different projects into ballet. So there is the crop protection project, which is my PhD project, but then we also did something about a ballet on atmosphere and how global warming and different types of particles affect the way the atmospheric warming is happening. And also we did one on sort of a more social science and how community behaviors can have an effect on climate science overall. Now, today I'm going to tell you a bit more about my PhD research and how I put that into ballet. So my PhD research was pretty much spent at the microscope, and that means I had a lot of visual information to work with. So at the microscope, I looked at how a plant germinates, and I tried to reproduce that in ballet form. So this is a plant germinating, this is a bean, and it germinates with the first radicule, which I used the extension of the leg to mimic that. And then what emerges from the top here are the 2 cotyledons, which are the 2 embryonic leaves. And again, I used the arm movement to mimic that cotyledon germination. Then we move on to the spore, the pathogen spore movement, and I used these circular movements for the dancers interpreting the pathogens to mimic these sort of chaotic swimming behavior that you see here. And then finally, I tried to look at spore attachment by getting the dancers to attach one at a time to the actual plant and then grow in a sort of mycelial-like fashion within the plant. Well, now let me actually show you the finished product.